I'm going to talk a little bit about what you've heard from the CAR T cell perspective, and how we try in the lab to study the CAR T cells and then act to make them better. So like you've heard, the CAR Ts are basically, you take the patient's own T cells, and then what we do in the lab is basically we re-engineer them to be professional myeloma fighters. But the problem that we sometimes get is that those cells sometimes, when we infuse them back to the patients, they're able to kill the myeloma cells because they recognize a specific protein that's on those cells. What we're trying to understand in the lab is what happens in cases where this doesn't work well. What happens where, unfortunately, the disease comes back. And this sometimes happens because of, I would say, mainly 3 different reasons. So one is that some of the myeloma cells are trying to escape the CAR Ts by cloaking the target that the CAR Ts recognize. So that's one thing. The other thing is that's the main problem that we see, is that some of those CAR T cells, they're becoming exhausted. And they stop killing the myeloma cells, and they eventually die. And we can't really see them when we look again at the bone marrow or in the blood. And the third thing that's problematic is the neighborhood that the myeloma cells are living in. So the bone marrow is actually a very hostile environment to the immune system. And there are a lot of other cells there that we call suppressive cells that are basically that prevent the immune system from doing its surveillance and its natural role as a cancer killer. What we're trying to do in the lab, and with your, I mean, some of you maybe saw me lurking next to the biopsy room with ice, taking some bone marrow and blood samples. So we're doing that because we take those samples right away and then we process it. And what we're doing in the lab is we're taking the bone marrow and the blood cells, and we use what we call single cell sequencing. Basically, we take the cells from the marrow, the immune cells from the bone marrow and from the blood, and we take each cell individually, encapsulate it into an oily droplet, and then we're able to extract the molecules from every cell individually and sequence it with genomic technologies. Then we use a lot of big data approaches like machine learning and social network algorithms, and we analyze that data. And what we're hoping to learn is, first, how we can take those CAR T cells, and if they express some unique things when they're exhausted that we can target with a drug, with a small molecule, with an antibody. That's one thing. The other thing, we're looking at the environment that those cells live in, with other cells that they interact, and whether we can drug that. This is really, I think, getting our own CAR T cell program is really at the heart of this, because like Tom, Nina, and Jeff mentioned, I think in academia that's the natural place for this to happen, where we can have discovery along with combinational therapies that the pharmaceutical companies can't really do on their own. I think with that, I'll just let you ask questions. And thank you so much for having me.
So are there any questions for Guy?
So Guy, how many different types of cells are you looking at in the bone marrow?
So the short answer is a lot. So we basically, our analysis is basically focused on two main areas. So one, the T cells themselves. And when I mean the T cells, I mean the CARs and also the residing T cells within the bone marrow. Because some of those T cells were actually studied in the past, and we call some of them, for instance, T regulatory cells or other T cell types, were shown to affect, basically to also suppress the good T cells from acting on the myeloma. We're also studying the myeloid cells, the NK cells, dendritic cells, basically all the immune cells in the bone marrow that are next to the myeloma cells.
So this is what we refer to as the microenvironment in the bone marrow, because we believe that one reason these cells may not last forever is something's going on in that microenvironment. And we may give Guy 5 million cells to analyze, and he looks at all these individual cells, which is why we need big data. But he looks at them all individually. We have a question over here, Jean.
What do you mean by exhausted cells?
It's a very good question. So exhausted T cells are actually cells that were stimulated, so they secreted some molecules that help them to kill other cells. But then at some point they become tired. They say, okay, I can't do this anymore. I need to rest. And I mean...
We don't understand why these cells get old,
Yeah.
why they just die. And in fact, one way in which Celgene's trying to approach that, or Bluebird's trying to approach that, is to try to pick younger killer cells. But they're going to age as well. So we need to develop, amongst our scientists, a way to make these cells immortal. And so they don't get exhausted, Gene. And that's one of the ways in which UCSF is trying to approach this problem. Thanks, Guy. Oh, I'm sorry.
Just wondering what the time scale is. Is it minutes, hours, days, years?
You mean the exhaustion?
Yeah, right.
So it's very patient specific. A part of it is we feel that it's because the current trials were being performed on patients that have seen a lot, right? That went through numerous regimens. So we're hoping in the near future to get this to an earlier stage of therapy where those T cells might be healthier and might be less exhausted. That's one thing. And the other thing that, like Jeff mentioned, is that maybe we can pre-select specific T cell types and then engineer those cells and infuse them to the patient. We typically see that within, I would say, weeks to months.
So a better way to look at that, actually, a more practical way, is we have patients who achieved, even quickly, within weeks or a month, a complete remission where we can't measure the disease at all. And many of you know how interested I am in minimal residual disease measurement. These people get to an MRD measurement of 0 in a million malignant cells. And then, for some reason, a month later, those T cells, those CAR T cells disappear and the disease comes roaring back. So we don't know why they disappear, why they get exhausted. Maybe they need continual stimulation, and there's some way to do that. We have Wendell Lim at our institution is looking at ways of turning on and turning off these cells. If they're too active, turning them down. If they're too, with just a molecule that you give the patient, a tetracycline pill or something. And then in ways to turn them up when you really need them again. So this is still in the future, but that's what I mean by a platform. The therapy is a platform. We can go further by making these cells live longer. That would be the next step. But another way to do that is to find a new target. Right now, all these therapies, you're going to hear from Tom and Debbie later, who had a BiTE, and you'll learn what a BiTE is. All the targets right now in myeloma seem to be at a BCMA, B-cell maturation antigen. Well, maybe there are better targets, and that's what Arun Witta is going to be talking to us about, how we can maybe find better targets for our cars and our bites. Arun?